students, today we are going to continue the remaining portion of the chapter cell components. In last class we have learned about the prokaryotic cell, and today we are going to understand something about eukaryotic cell. Regarding the size, we know that size-wise it is bigger than prokaryotic cells. And next, coming to the plasma membrane, all eukaryote possess the plasma membrane. Next, regarding the cell wall occur only in the plant cells in the case of eukaryotes. And what are the peculiarities? Peculiarities of the cell wall. It is a dead and rigid covering, and also it is composed of carbohydrates, cellulose, pectin, hemicellulose, and lignin. These are the materials that form the cell wall. And for the cell wall, 3 layers are there. A middle lamella and primary cell wall seen below the middle lamella, and below that again a secondary cell wall or plasma membrane. These are the 3 layers of cell wall. The other thing that we see in the cell wall is the plasmodesmata. What is plasmodesmata? Plasmodesmata means it's a pore that is seen between two contacting cells. It is a communication opening. Through that opening, the protoplasm from one cell can move to the other cell. That's a plasmodesmata. Next, nucleus. Nucleus, we know that in the case of eukaryote, nucleus is covered with a nuclear membrane, and also it is porous. So many pores are present, and because of the presence of these pores, it provides some provision for the entry and exit of molecules and substances from the nucleus and also towards the nucleus. What is the significance of the nucleus? That's nuclear membrane. The significance of nucleus is that it encloses the chromosome, right? In the case of prokaryote, this nucleus, this covering is not present. In that case, it exists in the form of a nucleoid. Then inside the nucleus, nucleoli, it is not shown here. Anyway, another spherical structure called nucleoli is present. And what is the role of this nucleoli? To produce the ribosomes. Next, we are going to understand about which are the cytoplasmic structures. Inside a eukaryotic cell, there will be double membrane bound organelles and also single membrane bound organelles. Then which are the double membrane bound organelles? They include endoplasmic reticulum, Golgi complex, mitochondria, and chloroplast. These are all double membrane bound organelles. Then which are the single membrane bound organelles? They include vacuoles, mainly vacuoles. Vacuoles are the storage structure for starch, glycogen, fat. Then some non-membranous organelles also present. Non-membranous, I will correct it. That's best. It is the non-membranous. There's a pen. Can't see it. No, okay. It is coming. N, not N. Non-membranous organelle. Non-membranous organelle means it includes ribosomes. Ribosome is a non-membranous organelle. And next is cytoskeleton. Cytoskeleton are some skeleton-like structures that give shape, support for the cell. Then centrioles. Centrioles are usually seen very near to the nucleus, and it is present only in animal cells. and its role is to help during cell division. Then chloroplast is present only in plants. That's another difference also when comparing plant cell with the animal cell. Okay, next we are going to see a comparison of prokaryote with eukaryotes. It's very important. This can be asked for your examination. On which all characters, based on which all characters we are finding the difference between prokaryote and eukaryote. That is mentioned here. Regarding nucleus and nuclear membrane. In the case of prokaryotes, we know that or we already learned that membrane bound nucleus is absent. Instead, a nucleoid is present. But in the case of eukaryotes, nucleus is present with a nuclear membrane. Then with regard to chromosomes, in the case of prokaryotes, there is no two chromosomes. Only naked nucleic acid is present. But in the case of eukaryotes, it is well organized in the form of DNA and proteins also present. Then cell wall. Coming to cell wall, prokaryotes. In the case of prokaryotes, cell wall is present only in bacteria, but that cell wall is not formed of cellulose. In the case of eukaryotes, cell wall is present only in plant cells. Then membrane bound organelles. Membrane bound organelles, it is totally absent in prokaryotes, but in the case of eukaryotes, membrane bound organelles are present. Ribosome, in the case of prokaryote, it is 70 S. In the case of eukaryote, it is 80 S. And subunits are of 50 S and 60 S in the case of prokaryotes. In the case of eukaryotes, the subunits are 60 S and 40 S. Centrioles, it is totally absent in prokaryotes, but in the case of eukaryote, it is present in animal cells, not in plant cells. Flagella, very small flagellas are present and it is composed of one fiber. In eukaryotes, flagellas are present, is very large and consists of microtubules in the pattern of arrangement. 9 plus 2 arrangement. 9 plus 2 means 9 units are there arranged in the form of a circlet, and each unit composed of 2 tubules. That's 9 plus 2 pattern. Then cell division. Cell division is amitotic. Amitotic means, we'll draw here. Suppose this is the cell nucleus and it forms some invagination, develops an invagination, and that invagination continues and then meet together in the center. By that way, divide the cell into two. The main important thing regarding the amitotic division is that nucleus is divided equally, but cytoplasm is not divided equally. That type of division is present in the case of prokaryotes, but in the case of eukaryote, very correct, precise way of cell division is present, that is by way of mitosis and meiosis. Then exocytosis and endocytosis. Exocytosis means transport of materials out of the cell. Endo means transport of materials from outside to the internal side of the cell. It is totally absent in the case of prokaryotes. In the case of eukaryote, it is present. Then gene regulation. Gene regulation in the case of prokaryote, that regulation happen only at the level of transcription and with the help of operons. What are operons? I will teach you later. There are some method of regulation how in what way or which gene need to be expressed at a particular time. That all determined by these operons. Then in the case of eukaryote, a different level of gene regulations are present, not only in the case of transcription level but also in the translation level also. So the comparison. By this way we finish this chapter in today's section. Thank you.